So when DNA polymerase introduces these mutations, there needs to be a mechanism for it to fix them. And the first line of defense, so to speak, is what's called this 3' to 5' exonuclease activity. And we talked about this briefly in the DNA replication chapter. Both prokaryotes and eukaryotes, their polymerases have this exonuclease activity. So in bacteria, it's polymerase 1 and polymerase 3 subunits. And you can see here, this is our RNA primer during replication, and then the bases are being added onto the 3' end of the growing strand, and this is a location where the DNA polymerase inserts an incorrect nucleotide. Now, if we have what's called 3' to 5' exonuclease activity, you can think, what direction do you think this polymerase is going to move when it's utilizing this activity? Well, in fact, it's going to move backwards. So this is an example of a polymerase moving not in the 5' to 3' direction, but in the 3' to 5' direction. This is the polymerase will backtrack, cleave out the incorrect nucleotide, and insert the correct nucleotide. This activity is inherent in the DNA polymerase, and this 3' to 5' exonuclease activity is generally pretty efficient. It works pretty well to ensure that the DNA polymerase is not inserting incorrect nucleotides. Now, there are times where this doesn't work, when there is an incorrect nucleotide inserted and the 3' to 5' exonuclease activity is not working correctly. This can occur if there is a mutation. For example, if there's a mutation in a eukaryotic gene for one of the polymerase subunits that codes for, or that possesses this 3' to 5' exonuclease activity. And this is how they determined the role of this activity in polymerases. So let's think about what would happen if this 3' to 5' exonuclease activity doesn't catch this mutation. Well, there is a second line of defense for mutations that are introduced by the polymerase, and that's what's called mismatch repair. So mismatch repair will correct errors that are still there after proofreading. So you can see here, this is a replicated DNA strand, and so this is the new strand here, and the polymerase introduced an incorrect base. It's mismatched, there's not correct hydrogen bonding, and it did not fix it by its exonuclease activity, so the entire strand was replicated. The way that this works in prokaryotes is that there is this complex, this mismatch repair complex, that can determine which is the old strand and which is the new strand. And the way that it does that is that this old strand, you can see, has these methyl groups on it. So CH3 you might recognize from chemistry. This is a methyl group. And certain bases in DNA can get methylated. And when the new strand of DNA is synthesized, the methylation is not present yet. So there are enzymes that will come, and a certain amount of time after replication will methylate this new strand. So right after replication, after the DNA polymerase has inserted an incorrect base and has finished its replication, this complex comes in and recognizes that there's an incorrect base and looks at which strand is methylated. When it sees that there is an incorrect base and it can determine which strand is methylated, it cuts out a large part of the new strand, including the place where there is an incorrect base, and it inserts a primer. It undergoes the polymerization with DNA polymerase, so it inserts a primer, it introduces the correct complementary bases, and it ties together the part before and the part after with DNA ligase. So DNA ligase forms the phosphodiester bonds to make this a continuous DNA strand. So again, this this complex comes in, it unwinds the DNA, it cuts out a large piece of the DNA, it, uh, it will make a new stretch of DNA with DNA polymerase, and then it will tie them together with ligase. So all of those steps, all of these activities are present in this complex. Now this is in prokaryotes. In eukaryotes, DNA strands are not methylated. So it's actually not currently known how the mismatch repair complex in eukaryotes identifies the strand that is the old strand and which is the new strand. So this is a very, very important complex, this mismatch repair complex, because remember, we don't want mutations to occur during replication, and that's why the polymerase has 3' to 5' exonuclease activity. But even that fails sometimes. And then the next line of defense is mismatch repair. If this doesn't work properly, you're in trouble. This is actually, there are certain types of colon cancer. There are two types of hereditary colon cancer. One type is called hereditary non-polyposis colon cancer. You'll oftentimes see it as HNPCC. And there's been a lot of work to look at what are the mutations that in colon cancer, in this type of colon cancer, what are the mutations in genes that oftentimes lead to the development of this disease. And what you can see here is that there are these different genes, MLH, MSH2, MSH6, PMS2. These mutations are found in almost all of the, or at least in most of the non-polyposis colon cancers. And these are genes that are code for the proteins for this mismatch repair complex. So this is here is the prokaryotic version of it, mut-S, mut-L, mut-H. Eukaryotes, they have different names, MSH2, MLH1, MSH6, but they carry out the exact same process. And in this type of colon cancer, most people who have this type of colon cancer will have mutations in these genes. And that the reason for that is because if there are mutations in these mismatch repair genes, these mismatch repair proteins can't remove incorrect nucleotides, and these mutations will build up in the genome. So each time the DNA replicates and a wrong base is inserted and it's not recognized by the exonuclease activity, there's no mismatch repair to fix it. So far we've talked about just one type of spontaneous mutation, which is the introduction of incorrect base pairing by the DNA polymerase. So this can be tautomeric shifts and these non-Watson correct base pairs. There are other types of mutations that occur as well that are spontaneous. So we're still talking about spontaneous mutations and mutations that are not induced by an outside source. One example of this is what's called depurination. And this is when on the DNA a purine is lost. A similar other type of mutation is called deamination. And this is the removal of an amino group from the DNA. And we're going to focus more on deamination. This occurs a little more often and is a little better understood. So the way that deamination works is, and we talked about this a little bit when we talked about the structure of DNA, you can see that the cytosine has this structure right here. So we have an oxygen coming off the 2' carbon. We have an amino group on the 4' carbon. Deamination is when the amino group on this 4' carbon gets removed, and now there's an oxygen in its place. So the difference here, and you can see now that there's an NH on the 3' carbon due to the change in the hydrogen, the change in the bonding between the 3 and the 4 carbon. You can see here now, when cytosine loses this amino group, this is now a uracil. And this is not good because uracil is the equivalent of thymine in DNA. So essentially, and when uracil is inserted into is when uracil is present in DNA after deamination, this what this results in is the DNA replication machinery treating uracil like a thymine, and will put in an adenine opposite from it during replication. Another deamination event that can occur is on what's called 5-methylcytosine. And we'll talk more about 5-methylcytosine when we talk about epigenetics. But this is, again, a cytosine. Cytosines can have a methyl group on the 5' carbon of their nitrogenous ring. And this methyl group plays a role in regulating how genes are expressed. So when DNA is methylated on this 5' carbon, this generally decreases the expression of genes, particularly when this methylation is occurring in the promoter cytosine genes. And many of these cytosines are methylated in the genome, and they play an important role in making sure genes are expressed correctly. But when this amino group is removed from a 5-methylcytosine, again, we get deamination. Now this is a thymine. So you can see uracil and thymine, very similar. Difference being this methyl group here. So these two methylation events result in a cytosine being deaminated into either a uracil or a thymine. And this is a mutation. It's a different nucleotide that is now present in that location. These 5-methylcytosines, these are oftentimes referred to as what are called mutational hotspots. So deamination of 5-methylcytosine occurs much more readily than deamination of a normal cytosine. So this is a much more common event, and there are ways to measure how often mutations are occurring in the genome based upon looking at the conversion of 5-methylcytosine to thymine. So you can measure these mutation events, and there are certain parts of the genomes where these mutation events occur much more often. And this normally, most of the time, this occurs in parts of the genome that don't code for genes, but sometimes this occurs in coding regions, in regulatory regions also. So this can be a bad thing. This particular mutation is fixed by a process that's called base excision repair. Sometimes it's called BER. So